speaker is Professor Alberto Audicchio. He is Professor of Medical Genetics at the Department of Advanced Medicine at the University of Naples, Federico II. And he's also Principal Investigator in Skeleton.
Sí, ¿no?
He's going to present a presentation. His presentation is entitled is Safety and Efficacy of AAV-mediated liver gene therapy of mucopolysaccharidosis type 6. Alberto?
Thank you very much. I'll actually take it a little bit from far in time when we started to work on this very interesting project, which aimed at developing a gene therapy for MPS 6. So I will discuss first the preclinical data, and then we'll go to our very recent clinical data from the phase 1/2 trial. So as I said, this is probably it's a decade-long project that originated in my lab at TGEM in Naples, and that had the objective of the well, let me see if I can go. Where are we? Okay, that had the objective to find an alternative therapy for this rather devastating mucopolysaccharidosis, which is Marotolami syndrome or MPS 6, which results from deficiency of a lysosomal sulfatase, sulfatase B. And this deficiency causes a widespread accumulation of glycosaminoglycans, in particular dermatan sulfate, in many tissues and organs, including liver, heart valves, bone and cartilage. This is there are major skeletal abnormalities in patients with MPS 6, as well at the level of the cornea within the eye. But unlike the other MPS that Fatima mentioned, well, the Sanfilippo syndrome that Fatima mentioned in her presentation, there is no primary cognitive impairment in MPS 6, and in this is quite similar to MPS 4A. There is a standard of care for MPS 6, which is enzyme replacement therapy, which is based on the principle that recombinant enzyme can be infused into the bloodstream of patients and can be uptaken by the mannose 6-phosphate receptor, and therefore deficient cells are cross-corrected by the enzyme. And this is the same principle that we thought to apply in the context of liver gene therapy as an alternative strategy for therapeutic strategy for this condition. And liver-directed gene therapy is being considered for many inborn errors of metabolism because the liver is the affected organ in these conditions. But it's also interesting because hepatocytes are well suited for systemic delivery of therapeutic proteins, and therefore you can use liver as a target also for condition in which liver is not directly affected, hemophilia being one of the most prominent example of this. And one of the advantages of directing gene therapy to liver is that liver has a peculiar fenestrated structure, which allows exposure of hepatocytes to viral vectors that are delivered intravenously, and therefore transduction results rather efficient after intravenous administration of a viral vector. And you can probably you probably cannot read this in detail, this list of clinical trials of liver gene therapy for, again, both conditions affecting directly liver or condition in which liver is used as a target organ for secretion of systemic proteins. What I can tell you is that most of these trials use intravenous AV as a strategy to target liver. And so this is definitely an approach that it's widely investigated and under clinical evaluation. So the principle is that a single intravenous administration, and in this the approach is rather different from enzyme replacement therapy, which requires weekly infusion of enzyme, that a single intravenous administration of an adeno-associated viral vector, so the vector that we and others are considering is the same type of vector that Fatima mentioned in her presentation, targets liver, and upon this transduction, liver is converted in a factory for systemic secretion, in this case of a lysosomal enzyme. So it's a rather simple approach based on a single administration. And over the years, we have tested this approach in various animal models of MPS VI, from mice to rats to cats, and shown that the enzyme produced by liver, transduced by viral vectors and secreted into the bloodstream, is able to correct the disease phenotype in these animal models. I will just mention a couple of data, of preclinical data, before moving forward into translating this into the clinic. So the first data is actually the experiment that proves that if you provide only one AAV vector, if you administer only one AAV vector intravenously to young MPS VI cats, you can actually obtain long-lasting expression and secretion of arylsulfatase B into the blood of these animals. And these last stably for at least 8 years, which is the longest time point of observation in these animals that were then sacrificed. And the levels that you measure over time are actually using a dose of AAV vector that is used in a number of different clinical applications. It's actually in the normal range for all these years. So this really proved the principle that a single administration provides a stable expression and secretion of lysosomal enzyme from liver in a large animal model. And to show that this is actually effective, I'll just briefly show you some video of affected animals. And I will start actually with a control heterozygous, which would be like wild-type animal from the colony. This is a 1-year-old cat that is actually similar to any pet, 1-year-old pet. It looks like and moves, especially moves like a normal cat. And this is very different from a 1-year-old MPS VI animal from the colony, which has a peculiar phenotype. You can appreciate shorter limbs. It has this flattened muzzle. It has also this very peculiar fur. And it's also quite impressive how little mobility as an affected MPS VI cat. While if you treat an MPS VI cat at 2 months of age, and then you look at the animal when he's 1-year-old, you cannot distinguish the animal from a normal animal, neither in its appearance nor in its ability to move around and catch objects, as you can appreciate. So these and many other preclinical data allowed us actually to secure fundings from the European Union to put forward a consortium, an international consortium, actually an intercontinental consortium, that had the goal to bring liver gene therapy for MPS VI from bench to bedside. So we had an American partner who was in charge of manufacturing and releasing the GMP, the vector to be used in patients, which is produced under good manufacturing practices in US, and this vector was then imported within the EU and released in Italy, where the vector was used in patients. We had to perform with this material formal preclinical toxicity studies to support the safety of this material, and also had to go through a rather cumbersome regulatory activity with the Italian regulatory agencies to get approval to perform this phase 1/2 clinical trial, which has been performed at the University Hospital of Naples in Italy. And this is a phase 1/2 open label dose escalation study with a single intravenous administration of 88 and encoding arylsulfatase B from the same liver-specific promoter that we've been using in all our preclinical studies, in 9 subjects that are 4-year-old with MPS VI, that are 4-year-old or older. And this is first in human, so the primary objective is safety. of intravenous administration of AV8, but we also looked at efficacy through biochemical and also clinical endpoints. And I will show you some of the data that we have recently collected from this trial. So I'm listing here the main inclusion and exclusion criteria of the trial. So patients had to be under enzyme replacement therapy for at least 12 months before they actually discontinue ERT and receive gene therapy. So we collect actually data baseline, which are the data under enzyme replacement therapy. And then during the observation after gene therapy, patients are withdrawn from enzyme replacement therapy. We had to exclude the patients that had the pre-existing immunity to AV8 because, of course, this would have impacted on the efficacy of the treatment. Just for those in the audience that are not aware, there are about 40% of individuals in the population that have encountered AV vectors and AV8 and have developed this pre-existing immunity to AV8. So this is actually one of the limitations of this approach, at least in its current version. And we also excluded the patient that, because of the ERT, had high levels of anti-resolverase B antibodies. So about 25% of our patient population, we collected over time about 40, where we followed up about 40 patients with MPS 6. About 25% of them were excluded because of this high anti-resolverase B level. So we enrolled 9 patients so far in the trial. Actually, the enrollment is basically closed. We are trying to enroll one last patient, but we're not sure we're going to be able to do it. And we enrolled 9 patients that are both males and females. You know, the age of vector administration is very variable. We started with an adult patient, a 29-year-old, and then we went in younger individuals. So we even treated 3 10-year-old patients and 1 5-year-old patient. So these are, to our knowledge, the youngest patients that have received a liver-directed gene therapy. Just to, again, make a point about this, liver hepatocytes undergo proliferation early in life, and AV is an episomal vector. So this is the reason we excluded the patients that were younger than 4 years, because we thought that at that age, the hepatocyte proliferation could have impacted on longevity of transgene expression from an episomal vector like AV. We also had the various genotypes that resulted in residual enzyme in most of the patients, but one that was also the one with the most severe phenotype. And lastly, we had the different follow-up. Of course, the first patient enrolled in the trial has more than 3 years of follow-up, while the last patient enrolled in the trial has less than a year in terms of follow-up, a little bit less than a year. So I will show you now some safety and efficacy data. So I'll start with the safety data. So the vector infusion was very well tolerated in all patients with no adverse event related to the IMP. You can see in this picture one of our patients that is being infused with AV intravenously, and while he receives this infusion, he does his homework on the computer, and vitals remain stable during the infusion, and so there was no problem associated with AV infusion. I'll show you now some efficacy data, and I'll start with the first 2 cohorts of patients. So we had 3 different doses. So the first 2 doses are depicted here. So we treated initially 3 patients with a rather low dose of vector, which is 6 times 10 to the 11 vector genome per kilogram. And in this first group of patients, you can appreciate that in 2 out of 3 patients, we observed shortly after the infusion of the vector a rise in serum resolverase B activity. These levels are between 10 and 25% of what you can measure in a normal individual. One of these three patients did not show detectable aryl V, serum aryl V at any time after the infusion. And if you look at the bottom graph, this is the second group of patients that was treated with a half a log higher dose of vector, so 2 × 10⁻12 vector genome per kilograms. We had 2 patients in this at this vector dose. We didn't see a real vector dose response in terms of increase in serum aryl V activity, yet these 2 patients had increased aryl V as a result of the infusion that was stable over time. Despite the expression of aryl V, of circulating aryl V in the range of 10 to 25% of normal, these patients had an elevation, a slow but continuous elevation in urinary GAGs after the infusion of vector. And so we had among our criteria that if patients went over a certain threshold of urinary GAGs after the infusion of the vector, remember these patients are without ERT, they had to go back to ERT. And so all patients in these 2 groups went back to ERT. And the ERT basically reduced the levels of increase of urinary GAGs that was observed before. Interestingly, though, these patients that went back to ERT had, if you look at the left part of the slide, had, of course, persisting aryl V in the blood as a result of gene therapy, because, of course, as we said, the expression of aryl V is stable. And so this, of course, gives an advantage to these patients in that if they don't perform weekly infusion of ERT, which happened to them during this COVID time, and so if they had to make longer intervals within 1 ERT infusion in the other, for instance, 2 weeks or even 3 weeks instead of 2 weeks, their urinary GAG levels didn't increase because of this staggered enzyme administration and longer interval. And this was clearly observed in 2 patients. So there are 2 advantages that I see in those 2 lower dose cohort of patients. One is that they have some baseline levels of aryl V that they didn't have before gene therapy, and that they can tolerate a longer interval between one infusion and the other of ERT than the regular schedule that is weekly infusion of ERT. Of course, this needs to be confirmed on a larger number of patients because this is only 2 of our patients. Let's move now to the patients in the high dose of vector. So they received the 6 × 10⁻12 vector genome per copies, and we have 4 patients treated at this higher dose. And in these patients, there was an increase of serum aryl V levels with respect to the previous patients in all of them. In particular, one of them, the one that you can see in the graph in pink, had peak levels of aryl V that matched the wild type dose of a normal individual. And as a result of these higher levels of aryl V in this dose cohort, you can also appreciate that the urinary GAG levels remained much flatter than in the other cases, and none of them has returned to ERT yet because of the effect of the treatment. So I will conclude by saying that the infusion of the vector was safe and well tolerated in 9 patients, in 9 MPS 6 patients treated in this trial. In 4 out of 5 subjects treated with the first 2 doses, so the low and what we call the low and intermediate dose of vector, serum active aryl V was detected at 10–25% of normal, but was not sufficient to maintain urinary GAGs pre-gene therapy levels, so at the levels that were under enzyme replacement therapy. The serum active aryl V in these subjects, though, was higher than the corresponding pre-gene therapy value, and urinary GAGs were stable even with biweekly enzyme replacement therapy infusions. Serum active aryl B in subjects with the high dose of vector are higher than in the previous two doses, are in the, as high or higher than 50% of normal, and urinary GAG increase was very modest in these patients. There was no clinical worsening post-gene therapy in the high dose cohort, which did not return to ERT yet. And of course, these numbers are relatively low numbers. So we treated a few patients at each dose, but more clinical data are needed to establish efficacy. So I will finish the presentation by mentioning the people who have contributed to this work, especially I'm referring now to the clinical part of the work. So I'm particularly thankful to the group of physician scientists who have been in charge of the trial, in particular Nicola Brunetti-Pierri, who has been the PI of this investigation at the University Hospital in Naples with all his colleagues. I'm also grateful to Rita Ferla and other members of my lab who have been supported this project for a number of years and have contributed to the trial as well by analyzing many data from patients. I'm particularly helpful to Stefano Zancani and his team at the Teleton TGEM Translational Unit for supporting this trial, and there are a long list of collaborators, local and international collaborators that have contributed at many levels to the trial. In particular, also the funding agencies. This work has been funded at the preclinical level by the Teleton Foundation. It has been supported to the clinic by the European Union, as I mentioned before, and we also had contribution both at the preclinical and clinical level by the Isaac Foundation in Canada and by the US National MPS Society. And finally, I'm particularly thankful to the patients who have volunteered in this trial. Several of them had to travel from other countries and had to, like Turkey, for instance, or Canada, or Northern America, and had to stay with us in Naples for several months because of the schedule that was quite demanding in terms of visit in the first months after the infusion. And with this, I thank you very much for the invitation and for your attention, and I'm glad to take questions. We cannot hear you, Fatima. You're muted.
Right. Sorry, I was saying that thank you for your nice presentation, but just very promising results. And I have some questions from the audience. Let's look at this first. I say, once I said, Dr. Auricchio, it is better to say that you have long-term expression versus stable, since your CAT data suggests slowing, decreasing levels over time.
Well, it depends. So there are, if we're talking about the patients, I think the question is related to data in patients. So there are patients that have long-term and stable data, and there are some patients, in particular, that one patient who had particularly high levels of aryl B in the blood, who had a decrease in the levels of aryl B over time. So it depends. There is some variability. I agree, but it's not true for all patients.
And they say that beautiful work. Is this dose you will use?
Right. That's a very good question. So I think we are close to a therapeutic dose, but we are not there yet. I think if you look at the data from the last cohort of patients, there are differences. So there is one that's responding better than the others. So I think this points to the fact that if we increase the dose of alpha log more, we're going to be in the therapeutic range. So our objective is actually to test one additional dose in this dose finding part of the study before doing a larger study with this higher dose. That's a very good question.
Now, I was saying also you need for redosing, if you plan to redose, probably yesterday Federico Mingozzi explained. Right.
I think that's very exciting work that Federico has presented, and his approach and others that are also under development may have the chance to tackle this very important point of both pre-existing immunity, because I told you that about 40% of our patients are not eligible to the treatment today because of this pre-existing immunity, as well as redosing in patients who have received a lower dose of vector and may benefit from a second dose of vector. Absolutely.
Okay, because other questions are also regarding all these neutralizing antibodies, and when are you going to start the next phase to increase the patient population?
That's also a good question. We don't know yet. So I've got to be very open on this. So our lot of clinical grade vector is basically, sorry, there is an helicopter flying by, so
we cannot listen.
That is making. Oh, you cannot hear it. Okay.
No.
So the current lot of GMP material that we've been using in this vector is running shortly. So that's why I said that we will probably be able to treat one additional patient, but that is going to be it. So the next dose will require a dedicated lot. So we are currently looking for, first of all, to find the funds to support this new campaign of production, and then to perform this additional dose. So we hope it's going to be a matter of months and not years, but we don't have any definitive plan on this yet.
Okay, thank you very much, Professor Auricchio. I think we have to
move on
forward. And thank you very much for the beautiful presentation. And now we have our last speaker in this session is Professor Jim Wilson. He's Professor of Medicine and Pediatrics and Director of the Gene Therapy Program at the Paramount School of Medicine at the University of Pennsylvania. Dr. Wilson, we're looking forward for your presentation. Thank you.
Thank you, Fatima, and thank you for the invitation from the organizers. And I wish I could be not in Philadelphia, but in Spain, but at some point we will be able to do that. Before I get started, I'd like to acknowledge the two senior scientists that have contributed primarily to the work. Christian Hinderer, who's a senior research director, an MD PhD at Penn, and Juliette Hordeau, who is also a senior research director at DVM PhD. And they were primarily responsible for the work that I'm going to describe today. What I thought I would focus on for my talk is addressing some of the issues of distribution of gene transfer into the central nervous system, sort of generically, and what data do we have that guides towards the most efficient way to do that. And Fatima did touch upon this subject as well. But one of our challenges in translational research has to do with scaling from small animal to larger animals. For most diseases, not all, the only animal model of a disease is a mouse model, in which would allow us to evaluate pharmacology or disease correction. I have a picture down at the bottom of a picture of a mouse brain. We generally move to non-human primates or monkeys to evaluate safety and to try to scale the dose. And one of our biggest challenges in scaling gene delivery from a mouse to a monkey or a macaque is the significantly larger size of the brain, which creates very difficult challenges, physical challenges of distribution of the vector. And then we move into first-in-human studies, as we've heard from both of our speakers and many others, to evaluate the safety in a biomarker assessment of gene transfer. But if you just scale and evaluate down at the bottom, one of the challenges is determining whether we can achieve the same level of vector distribution in a human that we can in a mouse. So we focused a lot on the route of administration for CNS gene therapy. The simplest, and one that is being evaluated for targeting the brain, is an intravenous administration. There also are data in several diseases, including Sanfilippo, where one directly injects vector into the brain through multiple burr holes to achieve broad distribution. The approach that we've been focusing on is delivery of the vector into the cerebral spinal fluid, either in a lumbar space or more proximal into the brain, into the ventricle, as Fatima had described, And also she noted another way to accomplish this is the cisterna magna, which is essentially a lumbar puncture, but below the occipital bone of the brain. Just a couple months ago, I reviewed the number of trials that are in the clinic now for delivering AAV to the brain for genetic diseases to determine what is the most popular route of administration. And the two most popular routes are either a direct injection, 7 out of 21, or a cisterna magna injection, 7 out of 21. So what is the biodistribution of AAV9 following an intravenous delivery into non-human primates for targeting the brain? Well, the challenge is for the vector to move from the vascular space where the vector is directed onto the other side where the neurons, astrocytes, et cetera, are located through this barrier, which is the blood-brain barrier. And Ron Crystal had done some nice work looking at imaging of the capsid following infusion into an intravenous delivery. And this is a sequential imaging of a non-human primate 1 hour, 24, 48, and 72 hours later. And again, this is an intravenous delivery where you see most of the vector deposits into the heart, lung, and the spleen. Also the thyroid because it's iodine labeled, with very little into the brain here. This is the distribution of the capsid as expected. But with that said, we and others learned many years ago that in larger animals, I'm going to show data in an MPS 7 dog, but cats and non-human primates, that when an intravenous delivery of AAV9 is administered, in this case the MPS 7 dogs, and then evaluated for gene transfer into the brain, cortex, cerebellum, or the spinal cord, that unfortunately not much delivery into the brain, but an amazing tropism for cells of the spinal cord, both the sensory and the motor neurons. So this suggests intravenous AAV9 at a minimum would be useful to diseases whose root are in cells of the spinal cord. And the one widely celebrated example of intravenous AAV9 targeting these particular cells is in the disease spinal muscular atrophy type 1, which is a very aggressive neonatal motor neuron disease in which children live really no more than 1.5 years to 2. The primary genetic defect is in the motor neurons that emanate from the spinal cord to the muscle. And these are the cells, coincidentally, that can be targeted by AAV9 when administered intravenously. And the group at Nationwide Children's, Jerry Mendel, Brian Kaspar, and then Avexis showed a remarkable success in an intravenous AAV9 delivery in these young kids with a severe disease, and in a product that was eventually approved called Zolgensma, which has turned out to be probably the most transformative commercially approved product, and one in which we all hope, those that have been in the field for a while, including my co-speakers, we can demonstrate similar results in other awful genetic diseases. So one practical issue that we considered, and this was Christian Hinderer actually during his PhD thesis, was how best to deliver vector to get broader distribution into the brain if the intravenous route wasn't as efficient as we need. So we considered two approaches. One is an infusion into the CSF through a lumbar delivery, or the cisterna magna delivery, as shown here, which is again a direct puncture into the subarachnoid space right below the occipital bone. And so why did we think that delivering vector into the cerebral spinal fluid would actually achieve broader distribution? And this is an area that I've studied a fair amount and not done primary research in, but did want to point out what the current thinking was about CSF flow dynamics. That the cerebral spinal fluid is a compartment that's found in the subarachnoid space, and there are the various ventricles, also the space around the spinal cord, and in the subarachnoid area. The pattern of CSF circulation is from the choroid plexus through the ventricles around through the subarachnoid space. The classic model of CSF flow was sort of shown here, where you have this subarachnoid space in between the dura and the pia, and then you have the brain, in which there was no contiguous or direct access from this space into the brain parenchyma, which would suggest that there would be little penetration of a vector when administered into this space. But over the years, this classic model has been revised, and here's an example of a study done about a little less than a decade ago, in which animals were injected with a fluorescent marker into this space and then followed over time. And they did show that the marker did provide access into the brain in what looked like perivascular spaces here. So this led to a model, as shown here, that when injected, a vector would be injected into this space, that there is a perivascular access penetrating into the brain, and then a diffusion from this perivascular access out into adjacent cells, and then eventually it exits the brain. So this would suggest that we can get penetration of the vector along these penetrating arterioles, and then adjacent to those arterioles, we could achieve gene transfer. We've studied the distribution of both an intrathecal, which is injection into the space around into the lumbar space, or an ICM injection. And what we think, what we will see is that in these penetrating arteries, that the vector would be able to distribute into deeper structures of the brain, and then adjacent to those arteries, we would have gene transfer. So the thought is that we could get broad distribution, but it may be in these sort of foci around the vessels. Again, this is the model. You inject a vector into the CSF, you have a penetrating vessel. The vector then distributes along the vessel and then provides access into the brain as it exits that perivascular space. And this is an experiment where we injected 1E13 genome copies of AV9 into the cisterna magna of a non-human primate expressing GFP, and then evaluated the animal for GFP expression. This is a high magnification view. So right on the surface, that has direct access to the cerebral spinal fluid, we get good transduction, but indeed the data would be supportive of this model, and that you see these areas here where there is deeper into the brain, these areas of transduction, providing a broad distribution throughout the brain, but not evenly distributed, because on a micro level, there's a higher density in the region right around the vessel. So we compared directly in non-human primates the biodistribution of vector when administered into the cisterna magna or into a lumbar space. The cisterna magna is red, the lumbar is in blue. And these are data which we published a few years ago, in which tissues from these animals, these are non-human primates, are recovered and evaluated for deposition of the vector genome. Again, blue lumbar, red ICM. These are brain structures, brain and spinal cord, and then non-CNS tissues, heart, lung, liver, and spleen. And what we see is that, as Fatima had noted in her studies, that vector exits the brain when administered into this compartment, into non-CNS components, but that we do achieve deposition of vector into both the brain and the spinal cord, but it's much more efficient when delivered in the cisterna magna than in the lumbar space. We did more limited studies in large animals directly injecting the vector into the ventricle, such as what Fatima showed, and our data were consistent with hers, that there was really no difference between an ICM injection and an ICV injection. So going back to the study that Ron Crystal had conducted, where he evaluated in real time the distribution of an AAV9 vector when delivered into the cisterna magna over 1 hour, 24, 48, and 72. And these are the relevant tissues, but the brain is located here and then the spinal cord here. So you see within 1 hour, there's broad distribution throughout the brain. And then within 24 hours, really throughout the spinal cord. But then over time, 72 hours, you see that the heart and liver start to light up, but not at all what one would have seen if one had injected vector directly into the blood. So the question is then, how can one use AAV for treating the neuropathic aspects of mucopolysaccharidosis and other like diseases? In many other talks, you would have heard or will be hearing applications of vectors such as AAV9 in these diseases, many of which were based on work that came out of our lab, but I'll just summarize kind of in one slide one advantage of this approach. And this is a study we did with Mark Haskins a few years ago, in which we injected an AAV9 vector expressing GUS-B into the, this was an ICV injection in an MPS 7 dog. And then we followed the animal for GUS-B expression in the CSF, and then 21 days later, we evaluated the animal for enzyme expression. And what we saw is that within 7 days, we achieved high levels of expression in the CSF that were stable. And we'll talk about the advantage of that in a minute. And then when the animal was evaluated, this is a dog model 21 days later, and evaluated for GUS-B expression in the cortex, cerebellum, and the spinal cord. In the untreated animals, there's no activity or no staining for GUS-B, but there is a pretty dramatic demonstration of not only transduction, but probably some cross-correction throughout the spinal cord and the central nervous system. That was within 21 days. And then when you look at a storage product, which is what is shown here, under normal conditions, we see nothing. Under untreated MPS 7, this is what we see. And then when an animal is treated 21 days before, we actually get complete clearance of all evidence of storage pathology. This may have an advantage in treating patients with infantile forms of diseases where the natural history is really quite accelerated and rapid, so that one can get full level of enzyme correction on board very quickly. So more recently, with a company that we started called Passage Bio, we've expanded our work in this area into GM1 gangliosidosis, in which there's a trial that's open and enrolling. Krabbe, again, a leukodystrophy, in which that trial is open and they're identifying patients. And then soon to follow would be MLD. But just to show you one example of the work that we did to prepare for these studies is a disease called Krabbe, which is due to a defect in this enzyme that leads to accumulation of a variety of byproducts, and as well as cytosine that may directly be toxic. So a classic storage disease in which there is a protein that's secreted. The natural history of this disease is most of those affected have the early form, the infantile form, and a very severe phenotype that they develop symptoms within a few months that include a variety of neurologic and motor deficits, fail to achieve milestones, and progressively lead to death within 2 years of age. In the United States, but not as much elsewhere, there is the thought that one could potentially treat these young babies with bone marrow transplantation to get a partial effect. But because the disease is so rapid that BMT, according to the criteria I understand, would be limited to those that would be treated within the first 30 days of life. This just shows the survival of untreated kids, showing how severe this is. And here's an example of some of the pathology of a young Krabbe patient that died at 15 years of age, showing these enlarged mononuclear cells and multinucleated gobloid cells that sort of characterizes the disease. So I won't go through all the preclinical work that we had undertaken, other than we teamed up with Charles Vitt from Penn to evaluate the efficacy of this in a dog model. And this is a dog that was untreated, and this is at 12 weeks of age. You can see the dog is losing motor function in its hind limbs. And a few weeks after that, the dog was euthanized for humane reasons. And this is a sibling of that dog, and this is at 19 months. Call this dog Oligo, and clearly no apparent motor defect. So we're quite excited about these preclinical data and look forward to rolling this clinical trial out with Passage Bio. But I want to point out just one other vignette and then end with maybe some speculation about the future, and that has to do with safety. And what's remarkable about AAV gene therapy is sort of how safe it really is and has been, although there have been more recently some evidence with high-dose AAV that there could be some severe toxicity. But we found in our first GLP study that we did with an ICM program, and this is with Regenix Bio and our program with MPS1, some pathology in a non-human primate that had to do with these structures. They're neurons in which the cell bodies are in these ganglia that are outside of the spinal cord, called dorsal root ganglia. And these cells have extensions of their axons up the spinal tract and then more distally into peripheral nerves. And we saw pathology in these primates who received vector by an ICM route. And this just shows an example of degeneration of these large cell bodies, and then these proliferation of mononuclear cells as well around those degenerating dorsal root ganglia. The axons then begin to degenerate, and you get these ghosts. We call it an axonopathy, which is essentially dropout both in the spinal cord and peripherally in the peripheral nerve of these sensory neurons. So what we learned is that when one injects a vector into the cerebral spinal fluid, almost any vector, one of the most highly transduced cells are the dorsal root ganglia. And here's an example of a monkey in which we evaluated the dorsal root ganglia following infusion of vector. And these are the large nuclei evaluated by in situ, and you can see extraordinarily high transduction, not only by RNA, but by protein. I think this was GFP. So that's good if you want to treat a disease whose root is in the sensory neurons, but we were worried that maybe that very high transduction had a negative impact on the viability of the cells. So we basically spent a lot of time studying this, and from a non-clinical perspective, we've conducted studies in literally hundreds of animals, multiple studies, and we are shown that it really is a function of the platform that we really see very consistently in virtually any animal treated by an injection of vector into the CSF. The pathology, although with some rare exceptions, it's asymptomatic, although we can measure the degeneration of the peripheral nerve using nerve conduction velocity measurements. Clinically, what's remarkable is that there's been little ascribed to this potential finding in humans, except there was a patient with ALS that received an RH10 vector into the lumbar space that did develop very soon thereafter evidence for this peripheral neuropathy, including severe neuropathic pain. So in order to try to understand this and help mitigate it, we developed a hypothesis as to why there is degeneration of these dorsal root ganglia. And what we're currently thinking is that they're so highly transduced that there's a significant load of vector that leads to stress on the cell, and then eventually it's degeneration. There could be a component of immune toxicity, but generally there isn't because it's self-limited. And so if that's the case, we needed to determine whether it was too much capsid DNA, too much transgene RNA, or too much protein. And we speculated that it may be expression, not necessarily capsid or the DNA. And so Juliet developed a strategy to specifically extinguish expression of the transgene of an AAV vector in the dorsal root ganglia without affecting expression in other tissues. So, so the approach that she took was based on the observation that that in the dorsal root ganglia, there's a microRNA called 183 and 182 that's expressed at very high levels that is not present in other neurons generally. So if that were the case, maybe we could use this as a method to specifically suppress expression in dorsal root ganglia. So what she did is she cloned into a vector, and this was part of our an MPS1 project, targets for this this microRNA, so that if this vector ended up in a DRG neuron where the microRNA was expressed, it would shred the RNA and decrease expression of the protein. But in cells that didn't express this microRNA, the presence of this target would have no effect. So we've published these results, but I just want to show you the data. What we did was we incorporated this microRNA into an AAV vector expressed in IDUA for a possible MPS1 application, and then injected this second generation vector along with the first generation vector and asked the question, is expression of the gene suppressed? And without the microRNA, high levels of expression in DRG, with the microRNA completely gone, but no impact on other expression in other cells. This just shows quantitatively suppression of expression only in DRG and not elsewhere. The question is, does it then help diminish the histopathology? We did a similar comparison of actually the first generation vector with the one that included the microRNA. There was this axonopathy here, but we were able to suppress it here. We also tried to use steroids to suppress this, which had no effect. And this just summarizes those results. So we are trying to, for all of our programs where we inject vector into the CSF, ask the question whether there's an axonopathy component, whether we can suppress it. So just in the last slide, where is the field headed? There's a lot of work trying to develop capsids that could traverse the blood-brain barrier and possibly affect a broader distribution of vector into the brain because of microcirculation, the capillaries provide access. And there is precedent or a lot of work in the ERT space and now vectors for penetrating the blood-brain barrier. There was a widely celebrated example of a vector that was developed that could achieve penetration of the blood-brain barrier, at least in mice, and it was an AAV9 variant, and showed that in normal mice, IV AAV9 did not transfer genes to the brain, but this variant, it could. It turned out that this was due to the recognition of a receptor on the endothelia of the blood-brain barrier called Ly6A, and Juliet made this discovery. But the problem is that this wasn't translated to other mice or wasn't translated to other humans. So maybe we can think about in next year's symposium or the year after, possibly next generation capsids that could achieve better delivery into the brain. But my own view, based on a lot of work we've been doing over 10 years, is it's really hard to find anything better. And often when you do, there are problems with manufacturing it. So for now, it's my view that the best vectors to use are the ones that we have in hand and the ones that you are hearing about in the various talks. So with that, I'll end my talk. Again, thanks for inviting me on this weekend to present before the community, the MPS community.
For this beautiful presentation, I think you have shown quite a study on the route of administration to reach the CNS. I think your last data, for the moment, there is no questions yet by the audience, and we are running a little late on time, but I think it was very interesting your last studies on dorsal ganglia toxicity. And I have one question. Since you don't see any clinical problem, do you think it would be necessary to include all these microRNA target sequences in the vectors in the future? Or you think that it's better maybe to reduce the dose, or with lower doses in administering inter-CSF, it may not be necessary, just not to find any problem?
Yeah. Yeah, so I guess it depends who you're asking the question of. If it's health authorities, the health authorities clearly believe that the risk benefit is such in many diseases so that one can tolerate this axonopathy, because many trials are opening when, in severe diseases. But there also may be a threshold. So for example, the Novartis application of Zolgensma and intrathecal delivery for SMA2 and 3, for which there is standard of care Spinraza, and in which it's not as severe as SMA1, that trial has been on clinical hold for quite a while. So I think it's really risk benefit, and our thought is the more experience we have, as long as we can share the data and people don't not share the data, that may de-risk the entire platform. And then I think, Fatima, what at this point probably what is best practice is just follow your patients at some point using nerve conduction velocity measurements, which we've shown is predictive of the very, very severe cases when you have clinical phenotype.
Yeah, no, very nice. Now we have two questions. You say, since many MPS disorders have retinal pathology, will the microRNA 183 mitigation approach negatively impact any retinal benefit from the gene therapy?
I guess it really would depend on what the target cells would be. And we are now trying to determine, because we don't get much gene transfer into the target cells of the eye when the vector is delivered systemically. But if you did target the eye, then that would be something you'd have to consider.
Right. And in other words, can you compare cross correction to the method you provided? I think you are carrying cross correction.
Yeah, the cisterna magna delivery achieves the same level of cross correction. It's more of a function of the enzyme, right, or the protein in the cell that one transduces. So I don't think there'd be any limitation on that. The microRNA shouldn't impact on cross correction on cells in which it expresses protein.
Okay, I think that we have to move forward because the session has to end, because it will begin the next session, and I will have to close it. And I would like to thank you, Dr. Wilson and Dr. Auricchio, for the wonderful presentation. I think it has been a nice session on in vivo gene therapy to treat MPS, and look forward to continuous looking the progress in this field. And let's move now to the next session, that is the third session on therapies, that will focus mainly on the results of gene therapy trials. And thank you very much for your attention to the audience, and see you in the future. Thank you.